have high school students coming here. Let me thank first the De La Pietra families, who are the ones who really finance these activities. And in fact, they allow us to invite prestigious people like Professor Ian Stewart to come and lecture not only to you, but to us and to the community in Stony Brook. And one of their conditions, in fact, is that the professor who comes should not only talk to scientists in the Simon Center or the departments, but there should be one lecture dedicated to the general public and one dedicated to high school students. Because this is probably a way of getting inspired on activities or on areas of physics, mathematics, biology, and so on, that could be good for your career. Well, Ian is a fantastic mathematician. He's, of course, a fellow of the Royal Society. He has all distinctions, accolades that you can look in his webpage. And if you are going to be studying mathematics, some of the textbooks he's written, you're probably going to be struggling through them in algebraic geometry, algebraic number theory, complex analysis, and so on. But he's one of the best expositors of mathematics and even physics for the general public. He's someone who is, loyal to Einstein's dictum, which is that you have to make things as simple as possible, but not simpler. And he always has the utmost respect for the intelligence of the reader. Sometimes people who talk to the general public, they are not necessarily so respectful, and then frequently they are really bad expositors. Well, this is not the case of Ian, and in fact he's going to tell you about something remarkable, which is tiger stripes and leopard spots. Please, thank you.
Thank you, thank you. Thank you. Okay, thank you all for coming. So I want to talk about animal markings, patterns on animals. And the object of the exercise is to convince you that math has a lot to say about this kind of very interesting question. I'm not going to take you through the equations or any of the details. I'm going to show you the pictures and try and tell you the story. So, famously, tigers are stripy. I actually once gave a lecture where we brought a live tiger into the lecture room. I'm not going to do it today. 6-month-old tigress, she was lovely. Yeah, so tigers are stripy, and leopards are spotty. Cheetahs are spotty, but they're different sorts of spots. That's a jaguar. And that's a clouded leopard. So big cats have these amazing markings on them. And of course, it's not only the cats that have very interesting patterns. You'll all recognize these animals. And tropical fish have beautiful patterns of stripes, lots of different ones, spots, other things. You can just find many, many pictures everywhere on the web of these markings. And seashells of various kinds. I mean, some of these are quite amazing. You look at these patterns and think, how did it know how to do that? And the question is, I'm not going to talk about why do tigers have stripes, why do leopards have spots. There are theories about the reasons why they evolved in that way, to do with camouflage and other things. But what is the biological mechanism that creates these patterns? And the person who made the first real impact on that question, wasn't appreciated much at the time, is this man, Alan Turing. He died in 1954. You may have heard of him. He's mostly famous for his work during the Second World War at Bletchley Park in England, decoding German encrypted messages. He also did work on artificial intelligence, but I want to talk about and early work on computing, theory of computing. He was an amazing man. But I want to talk about his work on animal markings, which was motivated by this exotic animal. Yep, you recognize this? You've seen these before? It's a cow, yeah? Yeah, so have these irregular black patches on an otherwise white cow. And Turing was trying to work out, how does this happen? And he came up with a mathematical scheme, and here is one of his pictures of a cow, basically, with various dappled patches. As far as we know, he produced this picture somewhere about 1953 plus or minus 1. And this was very early days of the computer. I mean, we have rather better computers now, but it's partly due to him and people like him that we have those. And in 1952, he published a paper which is increasingly seen as very important, called The Chemical Basis of Morphogenesis. Now, morphogenesis is the biological process in which animals acquire shapes and markings and patterns and turn into the familiar things that we see when we look at them. It's kind of what's on the outside of the animal, if you like. But we all know that deep down inside, everything runs on genes, DNA. And in Turing's day, that was only just beginning to be understood. So his theory kind of ignored the genetic side. The modern version will plug the genes in as well, as you'll see. So what Turing decided was it's something to do with chemistry. It's let's think of the stripes on a tiger. Those aren't laid down on the adult tiger. They are cryptically present on the tiger when it's still an embryo, before it's even been born. At a certain stage of development, patterns of chemicals are laid down in the cells in a kind of stripy way. And only later, as the animal develops, when the hair cells grow, that pattern tells the hair cells what colour they ought to be at that point on the animal. So it's a kind of two-stage process, a pre-pattern in the embryo, which has already got the instructions for creating the pattern that we see in the young and adult animal. And the colours come from proteins. They're protein pigments. So Turing at the time didn't really know what the chemicals were, so he invented a name for them, morphogen, basically meaning shape creator, pattern creator. And he wrote down mathematical equations for how morphogens work. And the crucial features are twofold. At each point on the surface of the embryo, the chemicals react together, like in a chemistry lab. You do experiments, you mix chemicals, and they do things. But the products of those reactions diffuse sideways across the surface. And that affects how the chemistry is working at nearby points. And then you put all that together and you see what happens. And if you take Turing's equations, you can either do some interesting analysis mathematically with the formulas, or you can stick it on a computer and see what it does. And if you stick it on a computer and see what it does, this is the kind of thing you get. And at the top we see spots and stripes, so leopards and tigers in rather, they're not quite the right shape, but you don't see a square tiger. And there are more complicated patterns that can occur, which we're seeing in the lower three pictures. They can get very complicated indeed. So this creature, the boxfish, has this very intricate, interwoven sort of pattern. It's like stripes that are changing their direction at random, but the spacing stays much the same. And the other picture here shows a calculated pattern on a computer from Turing's equations, which looks kind of like the boxfish. It's not identical, but it's got the right sort of characteristics. And there's a German mathematician called Hans Meinhardt who was interested in the patterns on shells. And he applied the Turing equations to the shell patterns. Now, the way the pattern on a shell appears is that the edge of the shell slowly grows as the organism deposits more and more calcite on the edge. And the pigments are built in by rules that depend on what the previous pattern of pigments was and the sort of near neighbours of points on the growing edge. And it's essentially the same as Turing's equation, but the pattern kind of grows as it's laid down. So here on the left of each of these pictures is a real shell. On the right is a computer calculation using Meinhardt's version of Turing's equations. And it's a pretty good match, yeah? It's quite convincing. And here's another one. So the background is the calculated pattern, the foreground, the shell itself. That's just a real shell. So this theory seems to match experiments, match reality, which is encouraging. And even this kind of very complex pattern, the math and the shells agree. This is actually an example of, you'll have heard of fractals, you'll have heard of chaos. This is a chaotic solution of the dynamical equations that Turing wrote down. So where are these patterns coming from, mathematically? Yeah, they're solutions of the equations, but the equations, if you look at them, basically the mathematicians will work with an infinite plane, which is not a very good version of a tiger, but it's good enough for this kind of thing. So you have an infinite plane, and the equations for the chemical reactions and the equations for the diffusion are the same at every point. The whole system is symmetric under any kind of movement of that plane. Naively, you'd expect the solution to be the same under all of these symmetries of the plane. In which case, all you get is a completely uniform colour, a bit like a lion. It's brown, and it's kind of brown everywhere. Lions are not famous for their stripes or their spots. But there's another consideration that comes into play, which says, yes, there are solutions which are completely uniform like that, but that's not always what you see. So let's just talk about symmetries for a second. This probably is familiar material to many of you. What's a symmetry? It's not a thing, it's a transformation. It's a way of moving something around. So I take this 6-petaled flower, I can rotate it through 60 degrees, more than once if necessary, and it still looks like the same flower. You'd have to put a little mark on one of the petals to see that it's moved. Or I can reflect it in different axes, which are the dotted lines in these pictures. You can flip it over. And there are 12 different operations of that kind, transformations, that tell you the symmetries. And Turing pointed out in 1952 that, for example, when an organism is growing, there is a stage at which you start with an egg and a sperm, and they fuse, and you get a single cell, and it splits, and it splits, and it splits. You get 2, 4, 8, 16, 32. And eventually you get quite a lot of them, and they form what's called a blastula, which is like a hollow sphere, which is kind of spherically symmetric, which is what you might expect. But then, as in the other picture on the right here, cells at the bottom, at one end of it, start to grow and fill in that cavity, and it's not spherically symmetric anymore. It is symmetric under rotations which keep the top and bottom in place. So you can spin it like that, but you can't interchange them. And this is called symmetry breaking. And Turing said, well, look, you carry on with this, you end up with a horse, or a mouse, or whatever, a tiger, which is not spherically symmetric. So something happens to persuade the biology, the cells, and also any mathematical model to break that symmetry that means it should be the same everywhere, or it should be spherical, or whatever is relevant, and create patterns. And he said this, okay, it's to do with the stability of the solution. So in physics and in math and many other areas, something is stable if when you move it a little bit, it moves back to where it was. If you've got a ball in the bottom of a bowl and you lift it up a bit and let go, it'll go to and fro and end up back where it started. If you flip the bowl upside down, put the ball on top, and it isn't one of those bowls with a ring round it, and give the ball a push, it'll roll down. So that's unstable. And if the uniform solution is unstable, then the system has to do something else. And that's basically what Turing said. But he said it in quite an amusing way, and this was in a scientific paper. He didn't use my picture, but this is what he said. Think of a rod that's hanging, it's like a pendulum, but there's a pivot, there's the rod, and there's a mouse on the bottom of the rod. So the rod is going to sit vertically. And then the mouse starts to climb up the rod. Past the pivot point, keeps going up the top. The rod's going to swing sideways because the mouse is now unstable. So let's see this. I like to do it with not the pendulum, but with a rod that's on a spring. So here's a rod on a spring. Here's a mouse. Yeah? Let's put the mouse on top of the spring. Well, if it's a fairly lightweight mouse and a strong spring, nothing happens. But if the mouse is heavy enough compared to the strength of the spring, it goes sideways. This is not quite sort of Disney-style animation, but it'll do. It can go the other way. And in a sense, the symmetry is still there. So what symmetry are we talking about? With a few minor details, this picture is left-right symmetric. Forget the tail of the mouse. And the spring, well, don't worry too much about the spring. But the important thing is there's a left-right mirror symmetry. The vertical state is where the solution has that symmetry. And when it flips to one side, you say, where's the symmetry gone? Because it's not mirror symmetric anymore. And the answer is there is another solution, which is the mirror image. So the symmetry is still there, but you don't see it in a single solution. And this is symmetry breaking. Now, in the plane, there are rather more interesting symmetries. There are translations where you take a shape and just move it without tilting it. There's rotation. There's reflection. And there's a thing called a glide reflection. Looks like that. And these all fit together to give what mathematicians call the Euclidean group, which is all the rigid motions of the plane. And you can use the theory of mathematical equations, dynamical systems with Euclidean group symmetry, symmetry of the plane, to predict the sort of patterns that you will see when symmetries break. So I'm going to do this on a bit of the plane, a rectangle. And one of the simplest things you see when the symmetry breaks is a pattern of stripes. Yeah? So on the left, it's a graph of the concentration of one of Turing's morphogens, if you like. When it's high, you get orange. When it's low, you get white. And on the right, in different colours, is the pattern of colours you would see. And it's stripes. And that's still got a lot of symmetry. You can slide it vertically and you wouldn't notice any difference. If you move it horizontally, you have to move it a whole stripe at a time. You can't just move it a little bit. So some of the symmetry that way has got lost. And this is what Turing realised. And if you want spots, the symmetry breaks in a slightly different way. It's a very regular pattern of spots, but we get this pattern of spots rather than stripes. So these are typical patterns for this kind of mathematical system, and the biochemistry is using those typical patterns. So people have done some more realistic experiments. You can take a cow-shaped region and ask what sort of solutions do you get. You can look at a part of the tiger which is fairly cylindrical or conical. It's a nice mathematical shape. You'll recognize that as the tail. And what you see is spots at one end, and as the tail tapers, you can sometimes get stripes. And there's a kind of theorem here, which is that a spotted animal can have a striped tail, but a striped animal can't have a spotted tail. Because roughly speaking, in order to get the spots, you start with stripes, and then the stripes break up. And there has to be a big enough space for that breakup to happen. And this is fairly true in the animal kingdom. And here are a few other pictures of the kind of thing. So here's the theorem. Spotted animal can have a striped tail, but a striped animal cannot have a spotted tail. Turns out to be not quite true, actually, but near enough. So we've got a spotted animal with a spotty tail. And that's a sort of, well, that's not even a real tiger, so let's get the real one. That's a fake tiger. But there is an example of a leopard with spotted body, and if you look at the very tip of the tail, you see the spots have merged together into stripes. There isn't room for them to be spots anymore. And so here's the proof of the theorem. Right. What else has stripes? Well, angelfish have stripes. And 1995 now, a couple of Japanese mathematicians looked at this angelfish. It's a tropical angelfish. It's the emperor angelfish. And you can see it's got stripes. And if you look closely, you can see there are a few places where the stripes split apart. A single stripe has a Y shape in it. And Kondo and Asai were applying Turing's equations to this fish, and the math told them that the stripes should move. They don't move down the fish, they move along the fish. And so that divergence where one stripe splits into two will actually move along the fish. And they thought, well, this is rather surprising. Does it really happen? So they looked at some real fish, and they found that for this particular different angelfish, you have a small one, there isn't room for many stripes. As the fish gets bigger, new stripes come in. But they come in quite suddenly because you have to have, let's say, 2 stripes, then 3, then 4, then 5. So you can't just insert another one into a gap without disturbing everything else. So from time to time, the whole pattern changes, new stripe goes in. And the graph on the right shows that process happening. Time runs from front to back in the picture. And then we go back to our emperor angelfish. And so what they did was they kept these fish in a tank, and about every 6 weeks they would take the fish out, photograph it, put it back. And when they did this, what they found was, and hang on, should be able to get a, no, yeah, doesn't matter. You can see in the pictures that you've got the real fish at the top in these pairs of pictures, and you've got the mathematical calculation at the bottom. And you can see, let's take the top one there, top right. If you look closely at the fish, you see this Y shape, and it moves along. So the stripes are actually moving, as the math predicts. And here's a close-up showing rather more clearly how that goes. Look at the Y shape there. It's moved to the right, and now it's actually moved off that bit of the fish altogether. And down below, we're seeing the same thing. And these are all over the fish. Go back to that original picture, you can see several places where these Y shapes are. And the prediction is those would move. So, not just angelfish. The plecostomus, which is something that people who keep aquarium tanks are very fond of. Very interesting looking fish. They have spots, they have stripes, and again, the spots and stripes move. Just wait for you to settle in. That's okay. Welcome to the lecture. Okay, I'll quiet down. Okay, I'm sorry to hear you had problems with your bus. So you've arrived for a lecture with a title about tigers and leopards, and you're looking at fish at the moment. And you're probably wondering what the heck is going on. So, quick summary, I'm not going to show the pictures, but what we've been talking about is that on, for example, tigers you see stripes, on leopards you see spots, you have these patterns of animal markings. What is going on in the biology when the animal is growing that produces those patterns? So I wasn't talking about why those patterns are there, but how do they get there? And in 1952, Alan Turing, who's famous for his wartime work decoding German messages, wrote a paper saying, look, it's to do with chemicals in the animal at a very early stage of its development, which react together, diffuse across the surface, and create these patterns in cryptic fashion. And later, when the animal grows, the protein pigments in the hairs that create the pattern appear at those locations. And then we moved on from tigers and leopards to angelfish, which are also stripy, and Turing's equations applied to the angelfish predict that the stripes on angelfish move. Okay? Now, you'll have seen these beautiful fish in aquarium tanks, very likely. The stripes do move, but they don't move fast enough for the naked eye to see. It takes about 6 weeks for them to move a millimetre or two. But there are experiments, and I think I can actually just go back and show you. Here we are. So we're talking about an angelfish. And if you look closely at this, although most of the stripes are just parallel, there are places where they split like a Y. And you can look at those places and those move. And these are some experiments. I'll show you the important one. This picture of an angelfish taken at 6-week intervals. It's the same fish. It lives in the tank. They take it out, photograph it, put it back. And here we see one of those Y shapes. Here it's moved along the animal. By this point, it's actually disappeared off the edge of that stripy region, and below was the mathematics. Okay. So we got to this. The other fish I was talking about, plecostomus, you have spots, you have stripes. The spots move as well. So the same prediction occurs. Now this is slightly surprising. You tend to think of these patterns as kind of fixed. Stripes on tigers don't move. Well, except when the animal's growing, the pattern changes a bit. But in fish it changes quite a lot. And you can even see cases where, okay, there's a saying that says the leopard can't change its spots. Well, this is the opposite. This is the saying would be that the tiger can't change its stripes. If you're a zebrafish, you can start out stripy and end up spotty. And these are pictures showing time-lapse snaps of a zebrafish, which it shouldn't be called a zebra by the time it's ended up spotty. So that comes from the equations as well. Let's not worry about this. There are lots of other markings on fish. And the theorists have this kind of explanation for nearly all of them. I want to talk about something else which is similar but apparently unrelated. There were two Russian physicists, chemists, Belousov and Zhabotinsky. And in 1950, Belousov discovered a chemical reaction where he actually had a chemical that showed colours, and the colour changed to show different concentrations of oxygen in the system. And the colour kept switching, let's say red and blue. It went red to blue, back to red, back to blue, back to red, back to blue. It was oscillating. And all of the chemists said that can't happen. Oscillating chemical reactions are impossible. So Belousov said, yep, look, here it is, look at it, I've got one. No, it's impossible. About 10 years later, Zhabotinsky rediscovered this, and it was still, the chemists thought it violated some basic physical rules. And eventually they realised it didn't. But that was why they thought it was impossible. And you can do this with 3 chemicals in a little dish. You set it up, and it creates these amazing patterns. And these patterns move. And there are 2 main kinds. There's a thing like, it's called a target pattern. It looks like the targets you use for firing bows and arrows for archery. Concentric circles, and the circles move outwards and new ones appear in the middle. And then there are spirals which spin, and they hit each other and merge together. And this is another of these reaction diffusion phenomena. It's another thing that Turing's equations can do. So here are the target patterns and the spirals. I don't have pictures of them moving, but you can find them. With a certain amount of preparation, you can actually do this experiment in a lecture and see it happening real time. Now, it's not just artificial chemical experiments that do this. Your heart does this. The normal beating of the heart involves a target pattern-like wave of electrical activity which moves across the surface of the heart and makes sure it contracts in a nice regular rhythm. If that breaks up into these spirals, you get fibrillation, and unless something is done quite quickly, the heart then beats irregularly, and this can be fatal. So it's to everyone's advantage that their heart knows to do one of these patterns and not the other one. And carrying on in this vein, I want to talk about slime mould. Slime mould is a lovely organism. It's an amoeba. It lives in the soil, but it forms colonies. And the colonies can move as if it was an organism. And they can come into a clump and create interesting patterns. So here is a picture of some slime mould amoebas coming together to form a clump. And then that clump can turn into what they call a slug, because it looks like a slug, and it moves as a single thing. So there's a life cycle whereby amoebas come together to form this clump, which forms a slug, and the slug moves somewhere else, and if it's dry enough, it stops, and then it puts up a stalk with a whole pile of amoebas in spore form on the top, and they dry up and blow away on the wind, and each one of those starts the whole process again. And this is how it reproduces and how it colonizes different areas. So this is just an amoeba. It's a very smart amoeba. Here's some pictures of the actual organism. Yeah? And it knows to form these spirals. This is an experimental picture of a spiral wave, and in fact that picture, the spiral wave moves. This is a wave of chemical action in the amoebas. How do they know to do this complicated stuff? Well, they don't know how to do it, but there are chemical signals that pass between them. And those chemical signals obey Turing-like equations. And the Turing-like equations give these patterns, and so the amoebas give patterns. And actually, slime mold, here's some more pictures of them. So, yeah. So, two mathematicians in Oxford, mathematical biologists, modelled the chemical signalling, and they get this kind of pattern. It's just like Belousov-Jabotinsky. Now, nowadays, biologists know all about molecular biology, biochemistry, DNA, all of that sort of thing. And so they don't just... One of the reasons they didn't accept Turing's theory initially was, what's a morphogen? It's just a name for a chemical. Which chemical? I don't know. Well, how can we do experiments? But now we know an awful lot of these chemicals and signaling molecules. So it's possible now to answer that question of what chemicals are actually involved in this process. Can you look at the details and relate it to Turing's equations or similar modified equations which might be a little more realistic biologically? And there are some nice experiments and observations recently. 2006, you've got a mouse. Mice have hair. Hair sprouts from follicles in the surface of the skin. Those follicles should be spaced apart, not in a regular pattern, but roughly the same distance. You don't want a great clump of hair in one place and none somewhere else, like me. Right? You don't want a mouse that's partly bald and it's got too many hairs in one region and too few in another region. And they discovered it's a Turing process that creates this regular spacing. And this is just telling you that there are certain signaling molecules which are well known to the biologists. And the other one, I mean, I would never have thought of doing this experiment, but these people were dentists. And so they were interested in the development of the mouth and where the teeth go, particularly, of course, being dentists, but also the whole development of the mouth. And, of course, you do your experiments and observations on mice or rats or things like that. So they were looking at the mouth of a mouse. And inside the mouth of the mouse are patterns of ridges. And they're like the stripes on the tiger. The pattern of those ridges is controlled by one of these Turing processes. So to sum up the story, we've gone from 1952, the idea that you get chemicals that react together and diffuse, and this creates patterns. The patterns can be classified using the mathematics of symmetry breaking, or you can solve the more complicated ones on a computer and see what they look like. It applies to tigers, it applies to leopards, it applies to tropical fish, it applies to the ridges in the roof of the mouth of a mouse. And you can all go home and say, what do we talk about today? Well, what happens inside a mouse's mouth. It works. And we can now underpin that with the genuine biochemistry that's going on in the system. So the mathematics and the biology have come together in a way that when Turing started this 80 years ago, could not have been envisaged. So we have Alan Turing and his cow, which was the inspiration for the whole thing. Apparently he wandered around where he was working, grabbing people, showing them his computer picture of these markings on a cow. He said, "Does that look like a cow to you?" And they'll say, "Yes, Alan, of course it does, yes." And so on. And nobody thought he was doing anything that was really of great interest. But Turing knew what he was up to. So, Alan Turing, and that's the end of the slideshow. And we have time for some questions. Thank you.
Thank you very much. Are there any questions or comments? No, that's interesting. You have? Who has a question? Raise your hand. Okay. There you go.
Oh.
Please, a little silence.
I'm just going to run back to the beginning and show some of the other pictures for the benefit of the ones of you whose bus broke down. And the only way I know how to do it, here we go. So, oh, come here. Yeah. Okay.
So you discussed the patterns that can be found in animals, right? The stripes of cheetahs and leopards that are relatively, or tigers and leopards that are relatively inconsistent. Do we think consistent patterns, stuff like the extremely consistent, like markings on cheetahs' faces, things like that, developed by the same sort of biological logic?
Yes, the mathematics is applied to an ideal version. So maybe a flat plane. You don't model all of the intricate details of the actual shape of the animal while it's growing. So the models that we're doing would be interacting with the different shape of the face, the bones, everything else, which will slightly change how the patterns work. So the same process will go on in two different cheetahs, but they won't be identical because of these slight differences in development. Yeah, so the theory is about an idealized version. I mean, the stripes on a tiger are much more, they're not just simple parallel lines. But I did show some of the mathematical pictures which are, I think you can sort of fit together. Here we go. The actual mathematical equation can produce all of these things and lots of other ones, depending on what extra conditions you put in, which would model how it goes. I mean, we can't yet predict for a given cheetah, observe the embryo, map out the chemicals, predict what markings it will have on its face. You can predict the general distribution of markings and the kind of thing, but I mean, people who work with cheetahs, each cheetah has its own markings. This is your point. Yeah. I mean, people who work with whales, the tail of the whale is different for each whale and so on. So it's partly the environmental conditions as it was growing up, but it's mostly the conditions of other parts of the animal interacting with the markings. Yeah.
You showed the ridge pattern that was used from the Turing equations.
Yeah.
Does that mean like texture of like?
Yes.
Could be applied with that.
That's right. It's not it's not markings. It's the actual shape of the mouth that comes in ridges. But again, the same idea. The cells that create the ridge are being instructed to do so by a stripy pattern, if you like. They know where to create the ridges. So the pattern is like a map, telling them where to put the ridges. and other things are coming in biologically to actually make it ridge-shaped. So cells in creatures, as they're growing and developing, they divide, they change their type, they move around. Cells move. And it's a very complex process, but it creates the shapes as well as the markings. And in that case, it actually is the shapes, as you picked up on. Very good. Okay.
Actually,
Yeah.
how did he calculate it without knowing the genetics?
You saw the formula?
I don't actually have them on the computer. It's a calculus formula. It's a partial differential equation showing how the rate of change of the concentrations of the chemicals in time, which is what controls the reaction at one point, also change in space, which is the diffusion side of it. So it's a partial differential equation with both space and time, and then the concentration is a function of space and time, and the equation tells you how that function changes in both space and in time. If that's and they particularly look for equilibrium patterns where after a while it settles down to something that doesn't change in time anymore, but it's still, where the stripes are orange and where the stripes are white, those are different concentrations of the relevant chemicals.
Any more questions or comments? I'm old and have a good eyesight, so right.
So I was wondering if genetics play like a major role in this. Obviously it probably does, but how does, like you said, the moving of spots and different patterns, is that based on genetics as well, or is that a completely different system?
Underpinning all this is the genetics. The genetics is, among other things, providing the recipe for creating the proteins that form the protein pigments. It's creating the signaling molecules that are used to get the reaction diffusion Turing equation working. So if you go one level down from there are various chemicals that do this sort of thing and say, where did those chemicals come from? That's all in the genetics. Yeah? But the message there is, there isn't something in a tiger's DNA that says put stripes here. There's something that says create these molecules, let them interact in the way that these molecules naturally do, let them do their own thing and they will form these patterns, and then the genetics comes back in again and picks up on that and maybe tells the cells to start creating hairs and so on. So there's this constant interaction between the information in the genes and free-running chemical or physical processes which don't have to be specified in the genetics because nature knows how to do that bit. They do it on their own just by following the laws of physics and chemistry. Yeah? But the genes are in there at every stage.
So is a lot of it just random chance, essentially?
There's a certain amount of random chance, yes, I think so. The biology is not neat and tidy like physics and very, very reproducible. An organism is growing in an environment. It's inside, if it's a mammal, it's inside the mother. Things that are happening in the environment affect the mother, which affects the growing organism. So it's random in the sense that there are external influences happening all the time, and they slightly change what's going on. Biological processes have evolved to be very robust so that it still works, but we don't all look alike. We all look like people. Yeah, we're all recognizably very similar. We're genetically quite similar, but the differences between us are not just genetics, they are other things, other random influences as well. Yeah. Nope.
Does the process for the formation of patterns differ between animals like the tigers where it's just kind of happening, or like a monarch butterfly where it's a competitive survival advantage?
Yes, there's there is a sort of common mathematical structure, which are the Turing equations or similar things. Those are probably too simple to be biologically totally realistic, but they capture a lot. But then in addition to that, the actual molecules that are involved are different in different kinds of organism. The evolutionary pressures that have led them to adopt this particular type of marking are hugely varied. For example, in butterflies you get what's called mimicry, which is, so you can have very nasty poisonous butterflies that you shouldn't eat, and you can have perfectly harmless ones that look very similar, and they're trying to pretend to be the nasty ones. And snakes are like this. A friend of mine had a corn snake, which has very bright markings, and it is absolutely like a very, very poisonous snake. The corn snake is completely harmless. Even if it bites you, nothing's going to happen. You can wrap it around your hand. They quite like that. They like the warmth. So they evolved those markings partly because they're already in the environment where snakes with similar markings that were poisonous. And that's just, I mean, that's one of the simpler examples, let's say.
Thank you.
Yeah, biology is very, very complicated. This is why I do math.
Okay, well, I'm getting my exercise today.
There was a young lady over here with a hand up.
There are a couple of questions. I'm not sure who was first. Who was first, you or you? Well, ladies first.
Hi. So I don't know if this is like a question that I should ask, but has anyone like ever researched or noticed any patterns in humans, like their hair or like birthmarks, like?
That's a very good question. I don't actually know the answer because I was mostly focused on the animal patterns. There must be some, but we are very uniform in many ways. I mean, there are some genetic conditions where you can have, I think particularly in women, skin colour can come in patches because of genetic problems. And one of them, the dividing line down the middle, particularly down the middle of the back, down the spine, what's happening on your left-hand side and your right-hand side can be very different. And this is, I think underlying this is a thing called mosaicism. They're mosaics. It's, I think, because of the difference that women are two X chromosomes, men are X and Y. The two X chromosomes in women kind of compete with each other. And cells in different areas of the body will get their genetic information from one of them or the other one, and those could be different. And very important in early development is the point at which the left-hand side and the right-hand side of the growing organism are differentiated, and they actually have different genes going on to get the chemistry working. And so it's not totally surprising that there is this dividing line down the middle. I don't think that's actually an answer to your question.
That's okay.
But yeah. But I don't know of anything that gives us stripy people.
One more question.
Yeah. So towards the end of your slideshow, you showed us like the blue spiral of the amoeba.
Yeah.
And you said it was like the chemical spiral. I'm just wondering how they image that. Is it like like small pattern on the cell?
Let me try and find it. Yeah, it was the blue thing with the spiral. Let's have a look. Nearly there. Yeah, that one.
Yeah, that one.
Okay. I don't know how they imaged it, but what they're imaging is the calcium concentration. So what you've got here is thousands of these amoebas, and they're sending signals to each other, and the signaling affects the level of calcium. And they have some imaging system, and I'm sure if you looked up, if you dropped this picture into Google and said, what's it a picture of, you would get the reference to the paper, which would tell you the imaging system they used. And it will be some sort of technical name that certainly wouldn't mean much to me, but it would tell you how they did it.
So it's basically just
Yeah,
like
I
a
mean-
big mass of amoebas, and then they have like some sort of process that makes the calcium.
That's right.
Yeah.
Each amoeba is creating producing chemical molecules, and they're spreading, diffusing out. Whether it does that or not is affected by the molecules it's receiving from the other amoebas. And if you looked at this, you wouldn't see any of this pattern because it's just to do with the chemical concentration. But if you have the right imaging equipment, you can make it show up. Well, if there's more calcium, it will show up. I mean, for all I know, they mixed in some chemical which is sensitive to the amount of calcium. So the whole process, the whole thing, I mean, even this is slime mold, yeah? The way this collection of slime mold seems to act as if it's a single organism at times is all to do with the effects of the signaling between them. Each individual one doesn't know what it's supposed to be doing. It just listens to the signals coming in and reacts and sends stuff out. But the way those signals work actually creates this very complex but organized cycle of behavior for it to reproduce. And the evolutionary reason is is to reproduce and spread to new locations. Yeah. Thank you.
Any more questions or comments? Yeah, there.
You mentioned that the theorem that a spotted animal can have a striped tail, but a striped animal can't have a spotted tail, is almost always true. Why isn't it always true?
Okay, because there's an assumption made in the proof of the theorem, if you like, which is that the way the chemistry is acting. Okay, so roughly speaking, the proof is if you have a large cylinder, the first thing that happens is that the pattern breaks up into stripes, but if the stripes are long enough, they will break up into spots. Okay? So if you have an animal with a big fat body, it can have stripes, or those will break into spots. If you have a thin tail and you've got a fairly wide stripe, there isn't room to break into spots. You can even think of the stripe as a big spot that wraps all the way around. Okay, now, why isn't that a slam dunk proof? Because the chemistry might be different and run in different ways towards the tip of the tail, or anywhere else for that matter. I mean, look at the pictures of the angelfish. They have all sorts of amazing markings in different areas. You couldn't come up with a statement that the stripes in one part would have to obey exactly the same laws as the stripes somewhere else. I can't remember what the example that disproves that theorem actually is, but there are some organisms where you have basically stripes on the body and then the tail, for whatever reason, has broken up into spots. So there are some hypotheses of the theorem which have been to do with the chemistry working the same way at both ends of the animal. Yeah? Okay, good question.
Yes.
Is there any examples of it in the plant world?
Plant world.
Yes.
Yes. I mean, the whole formation of patterns in plants is very interesting. But the sort of natural symmetries of plants are more to do with the rotational side. So, I mean, if you look at markings on flowers even, you will see individual petals can have stripes and the like. Of course, the most famous mathematics of plants, which is related to this, is the business of Fibonacci spirals, and Fibonacci numbers of petals. Probably everyone's heard of this, but if you look at something like a sunflower, there's a pattern of seeds in the head of the sunflower. You've got a big sunflower. And if you look at those seeds, you find they form systems of spirals. They're packed together. And it's like two sets of spirals, one going that way and one going the other way. And if you count how many of them there are, you tend to find Fibonacci numbers, which are 1, 2, 3, 5, 8, 13, 21, 34, 55, 89. Yeah? Where each is the sum of the previous two. And those numbers turn up a lot in plants and in the numbers of petals, because whatever is producing this pattern. And the theory is roughly that this is the optimal way to pack the seed's head together. So there's a whole pile of stuff on that. And it does have, it's not identical to the Turing equations, but there are some fairly similar equations to explain how that process works. Yeah.
My last question?
Yeah. You said earlier that the movement of the spots and the stripes on the animals are random. Is that because the environmental factors that they're exposed to are random and are not always the same? Or is there something fundamentally like stochastic about the Turing's equations, like Brownian motion?
Right. I think it's a bit of both. I think that's an excellent question. Any sort of external disturbance, even with an idealized mathematical model, if I write down the Turing equations on the plane, same formula everywhere, but then maybe slightly change the reaction rates at some places or anything like that, or kind of change the shape of the plane so that it's a bit bumpy, yeah, then the whole way things diffuse will be slightly different, and therefore the outcome of the pattern will be different. So it will be a mixture of, it'll still look like a pattern, but it won't be quite the same pattern. And instead of regular stripes, you might get irregular ones. I mean, the stripes on a tiger are not nice parallel stripes, but there are more sophisticated versions of the model which give things sort of not bad approximations to what you really see on a tiger. But yes, I think it's both internally generated and environmental. Yeah. Thank you.
Okay, maybe it's time to wrap it up. This shows you one example of, there was a famous mathematical physicist, Eugen Wigner, who in the 60s wrote a very interesting paper. In fact, the most interesting part is the title, which is the unreasonable effectiveness of mathematics in the description of the natural world. It is kind of remarkable how mathematics is the language that somehow explains or expresses the way that nature works. Let's thank Ian for such a wonderful lecture, and I hope to see you soon. Thank you very much.